Hello? Hello? Hello, can someone please unmute the mic and tell me, am I audible? Hello? You need to tell. Actually, I don't know, am I audible or not. If you will tell, you will respond, then only I have to proceed. Hello? Geeta, hello? Nikita? Hello, Nikita? So am I audible? Yeah, thank you for the response. So why you people are not unmuting your mic? Actually, you can unmute and you can discuss. This is the interactive section only, right? So if you people will not unmute the mic, so how you people will interact? Ma'am, in hostel, other girls might get disturbed. Okay, okay, no problem. And others, for others, Geeta, Payal. Okay, okay. Okay, we will wait for some 5-10 minutes. So if someone has to join, then by the time they will join. Okay. So actually, it will be very difficult if you people will not talk. So okay, we will start. It's already 6:06. So Actually, I want to take introduction from you people, but you people are not unmuting your mics. So basically, I will introduce myself Shivangi. So I will be doing the TA-ship for this course. So this course is like Professor Espirat is teaching this course. So actually, this is a very interesting course that is bioinorganic chemistry. So as this name indicates, so this is bioinorganic chemistry, and it is the not dips like it is not purely bio and it is not purely neither purely bio nor purely inorganic. So it is the combination of bio inorganic. So this is the interdisciplinary course. So my name is Shivangi. So I am doing the PhD from Indian Institute of Science, Bangalore. So I will be like TA for this session from Monday, like from 6 to 8. I will be taking your doubt sessions and I will interact with you people regarding any of your doubts regarding this course. So basically, I was thinking I have to interact in this class as these are interactive sessions. So, but for that, you people have to unmute your mic, or means it is like this is my question to you people: how you will ask your doubts if you will not unmute your mic. If you want, huh? So if you I think professor has taught you like first week lectures, and they have uploaded the first week lectures in which I think they have discussed about the oxygen transport, bioenzymes and molecules. So if you people have any doubt regarding the lectures or something, then you people can ask. I will try to answer your questions, but sometime if I will not be knowing the answer, I will tell you in the next class, or I will read and prepare, then I will tell you. So now you people, if you people have any doubt, then you can proceed, and it will be better if you people will unmute your mic. At least the people who can unmute your mic, no disturbance will be there. If you are in your surrounding, then you people can unmute your mic and introduce yourself. There is no need to hesitation. I am also a student, I am also like you, so you people can interact with me. So any of you want to introduce yourself? Okay, if you are not talking, then your wish. So so any doubt regarding the lectures? I think you people are listening that lectures. You people are attending this course. So any doubts from your side? I will be happy to clear if any doubt is there. Hello, Gajanan. You want to tell something? Gajanan, do you want to introduce yourself? If you want to introduce and you are like feeling hesitation to talk, then you can write. I will read from there. So I can see five names here: Payal, Nikita, Geeta, Nikita, and Gajanan. So any of you have some doubt or you want to introduce yourself, if you want, feeling comfortable in texting, then you can text. If you want to speak, then you can speak. Okay, okay, no problem. So, yeah, okay, no problem. So, I will continue my class. Ma'am, are you talking about which session? It's our first session. Yeah, I am talking about first session. Is I am taking first time your class. That's why I am asking for the introduction. It will be first time for you people to interact with me. So that's why I am asking for introduction. This is first class only. But you people are like attending that video lectures, na? That lectures they are sharing on NPTEL sites. That you are hearing, na? That you are listening, right? Why? Why no? It's bioelectrochemistry. Oh, it's bioinorganic, right? 1 minute. No, actually, I think I don't know. Means for me, I am doing the TA ship for bioinorganic chemistry. So, it's you are telling like it's the bioelectrochemistry class. I am doing the TA ship for bioinorganic chemistry. Can you tell me the course code for this? Any course code, if you will tell? Gajanan, do you know the course code? Any of you know the course code for this course? Sure, sure. I'm waiting. BP1040. Actually, I don't know. Some misunderstanding happened. I am sharing this. Okay, I'm sharing one window with you. Can you please check it out? 1 minute. Is my screen visible? Any of you, please, can you tell, is my screen visible to you people? Okay. So, yeah, so can you please check with this column? This is, I am teaching this course, Bioinorganic Chemistry, Professor S.P. Rat, IIT Kanpur. So I think last week Pyali Mahapatra has taken the class. So now this is Shivangi, this is me. So I'm taking the class for bioinorganic chemistry. So can you people please confirm like I am in the, so this is bioinorganic chemistry, right? Here someone has told it's bioelectrochemistry. For bioelectrochemistry, now there are two TAs, Nitesh and Siranjeevi. So I am not taking the class of bioelectrochemistry. So this is the class of bioinorganic chemistry. I think you people have some. Huh, then actually I also get confused. Okay, okay, no problem. Actually I confused. I thought I am in the wrong class. So okay, okay, no problem, no problem. So this is the bioinorganic chemistry. Okay, so I am teaching the bioinorganic chemistry. Okay, no problem. Fine, fine. So this is bioinorganic chemistry class. So I will show you the this also. So this is the course code NOC23CY15. So this is the bioinorganic chemistry class, which is teach by the Professor S.P. Rad from IIT Kanpur. So this will be 4 week uh, class. So these are the course duration, and March 26 will be your exam. And these are the three TAs which will take your doubt session for this class. So I think Pyali has taken your doubt session. I don't know about Somnath. So I am Shivangi. I am this week I will take this session. So so actually I introduced myself, but again some of people have joined now. So I am Shivangi. I am a PhD student from Indian Institute of Science, Bangalore. So I am doing TA-ship for this course. So I'm doing TA-ship for this course. So my research field is also bioinorganic chemistries, in which I am synthesizing some of the cancer drugs. So as you people know, there is like um, bioinorganic chemistry is not any hardcore course, like it is neither biology nor inorganic chemistry, but it is a mixture of bioinorganic chemistry. So it is basically like an interdisciplinary course in which we will learn about some of the biological aspects and some of the chemistry aspects. So in this course basically we will learn about the use of the inorganic elements in the biology. So so basically, this TA they have assigned for clearing your doubts. So whatever they will teach, so I have to take your doubts. So this will be the first class from my side. but you people might have interacted with other TAs. So maybe you have asked some doubt from them. So I want to ask if you people have any doubt. I was seeing the NPTEL site for this course. They have uploaded the video. That first week video has been uploaded. So you have to I think you people have attended the classes. So now I am expecting some doubts from you people, that you people ask some doubts. So I will try to clarify your doubts. So this is, I told you, this is an interactive session. This is not like I am teaching and you have to just listen. You people can unmute your mic and in any time, any doubt, anything you can ask. Or if you are feeling like not asking, you can text in this message box. I will read from there. So I will be happy to clear your doubts. And basically this is the first class, so I want, like, if you people will introduce me, then it will be very, because all of us are from different backgrounds. So if you people will introduce, then it will be very good, we will interact. So if any of you can unmute your mouth, like mic, and introduce, then it will be good. So any of you, actually I can't take, like, I don't want to take any point. If you want to unmute, then you can do. And Gajanan, now is it clear now? We are in right class, right? At least you people can reply or write, we are in right class. This is bio inorganic chemistry. Yes. Okay. Okay. Fine. So now, um, any of you have any doubt regarding this course? Any question or anything you want to ask from me? Any doubt? So okay, I think you people have no doubts. So uh, if you have no doubts, then I want to teach from my side. So so I will go. So I will share my slides. Huh. So this is bioinorganic chemistry. I told you, in which we will. Oh my God, this is very bad to write here. Oh, here it is very difficult to write. Okay, I think I need pen for this. Oh my God.
Hello.
Yeah, yeah. Hello.
Hello,
Ha
ma'am.
ha.
Am I audible?
Ha ha. Yeah, yeah. Tell.
Hello.
Yeah, yeah, yeah. You are audible.
Actually, ma'am, this is my first lecture.
Ha
So,
ha.
can you just give brief introduction? Like, how can I get started? I know it's like been 2 3 weeks, but
Okay, okay. Actually, this is like, okay, I will do like that only. So, okay, we will go through like first lectures only. 1 minute. Okay. So, um, are you able to see the slides?
Yes, ma'am. Yes.
Yeah. So basically, in this course, we will learn about the like we will learn about. Can I know your background from from which background you are?
Sure, sure. Ma'am, sorry, I forgot to introduce myself.
Yeah, yeah.
Myself, Gajanan. Currently, I am pursuing Masters in Pharmacy in Pharmaceutical Chemistry.
Okay.
Like,
So
I'm from pharma background, ma'am.
okay, okay. So you are doing PhD.
No, no, Masters.
Okay, okay.
Like,
So you
I'm
are
in
doing.
second year.
Okay, okay. Now, so okay, fine. Okay, so you are from like pharma background, so it will be easy for you. So there is no problem with this course. So okay, I am starting this course. So yeah, so in this course we will basically learn about we will basically learn about like which elements are present in the biological system. So in this we will learn about the inorganic elements which are present in the biological system like biomolecules. So basically in our human body many elements are present, but out of these carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur, these comprising 98% of the human body. And in human there is like 60% of oxygen. So basically oxygen is the most abundant element present in the human body. So if we will see, so these are the different elements present in the human body. So they are saying like if we will see carbon, hydrogen, nitrogen, oxygen, they are the organic elements. So if they are making 98% of the human body, so along with these organic elements there are different metals like iron, zinc, copper, molybdenum, cobalt, chromium, vanadium, manganese, nickel. So these elements are also present in the human body. So they are telling like life is along with organic, it consists of some of the inorganic elements. So these inorganic elements, like these elements are present in the human body at the at the like we will say biomolecules. So biomolecules are the enzymes which are present in the human bodies, and in these enzymes, in the active site of that enzyme, metals are present. So what are the enzymes? Can someone tell what are the enzymes? This is very basic. I think you can answer this. No. Okay. So enzymes are nothing but they are also the catalyst. But the difference between enzymes and catalysts are like enzymes are the biological catalyst, whereas the catalyst is like a general term in which like catalyst is a substance which can increase the reaction, but it is neither like it is not important that this reaction will be biological reaction only. So catalyst is any substance which can increase the rate of organic inorganic reaction outside the environment. But when we are using the specific term enzyme means it is a specific catalyst which is present in the which catalyze the reactions in the human body. So they are biological catalysts. Enzymes are basically biological catalysts. And the enzymes which contains metal in their active sites, they are called as metalloenzymes. Metalloenzymes means metal plus enzyme. So like in the active site of enzyme, metal is present, so they are called as metalloenzymes. So these are the different elements which are present in the uh, active site of the like enzymes. So these elements are necessary for our human body, and if these are present in the deficiency, or if these are present in the excess, so these type of like problems can happen. You all know that if iron content, we know that if like heme level is low, then we will call it as like anemia. So in the heme is also a enzyme in which iron is present. So basically if there is a deficiency of iron, then people will get anemia. And also if copper will be in excess, then wilson disease will happen. Like copper will accumulate in the brain of the human and it will leads to the wilson disease. And there are other like Menkes syndrome, which is due to the deficiency of copper. So these are the various diseases which is associated with the deficiency or the excess of the metal ions. So these are the biological elements, these are the biologically important elements. So these are the bulk biological elements we all know, like hydrogen, carbon, nitrogen, oxygen, sulfur, phosphorus, potassium, sodium. These elements are present in the bulk, whereas whereas these elements like gray with white, these are the elements which are essential, and these brown color elements are possibly essential for some of the species, not for all. So we all know that what are the proteins? Protein is this type of, like it is a, polymer with amino acids as a monomer. So in this proteins, amino acids are present. So amino acids are the repeating unit of the protein. So amino acids are the monomer, and if amino acids, if, like me like when there is a polymerization of amino acid through peptide bond, it leads to the formation of protein. So amino acid, this is the nitrogen amino group, and this is the carbon group. So if these increase, like if these polymers, like amino acid will polymerize, it will lead to the formation of protein. So So there are different proteins present in the human body. So enzymes, I told you, these are the biological catalyst, different hormones, different storage protein, transport protein, structural protein, protective protein, contractile protein. So these type of the proteins are present in the human body. So enzymes are the catalyst which basically increase the rate of the reaction. Like amylase, it begin digestion of carbohydrates. So if in body carbohydrates are present, so it has to digest. So amylase is the enzyme which catalyze this reaction. So hormones, these regulate body functions, and insulin is a hormone. So this is a storage protein which store, like which store some of the necessary things which is required for a human body, but not all the time. But sometime we will feel like exhausted or something, when we need something, then that kind of protein will activate. Like we know, this myoglobin is a protein which store oxygen in the muscles. So whenever we are feeling exhausted, like most of the time during exercise, so that time this myoglobin, that body will need oxygen, like that time myoglobin will give oxygen to the body. And these are transport proteins which carry substance from one body organ to the other body organ. Structural proteins are not the proteins which are directly associated with the human reaction, but it provides shape and support to the protein. So protective protein, it like in body different type of foreign metal can enter, so it defend the body from these foreign materials. And these contractile proteins like myosin and actin are the, best example for this contractile protein. They will do muscle movement and do mechanical work. So these are the different proteins which are present in the human body for various functions. So these are the structures. This is the, like, amino acid. So if it will polymerize, it will lead to the formation of protein. So proteins are also different time, like um, primary structure, secondary structure, tertiary structure, and quaternary structure. So if these amino acids are polymerize in a single linear fashion, then it will lead to the formation of primary structure. So here you can see that this is, I am assuming that you people know what is amino acid and what are the structure of amino acids. Shall I go in detail of amino acids also? Can someone tell, like, do you aware of that amino acid, right? Gajanan, can you please unmute your mic? You can tell, other people are not telling anything. Okay, okay. Okay. So Huh. Huh. So when when amino acids are joined in a linear fashion and they polymerize, they will lead to the formation of a linear protein. And then if there is no any complication in the protein, then we will call it as a primary structure, which will tell about the sequence of amino acids. Like this is an angiotensin, this is a protein in which asparagine, arginine, these are connected in a linear fashion through peptide bond. So this is the secondary structure. So secondary structure is like it is a helix like structure, it will like twist. So it is not linear, so it can twist right handed or like clockwise or anti-clockwise. So it can form um, uh, like alpha helix will form, and this is the beta sheet. This is also a form of secondary structure in which this is parallel beta sheet and this is anti-parallel beta sheets. So in parallel sheet beta sheets you can see the hydrogen bond in linear fashion, so these are the parallel beta sheets, and these are the anti-parallel beta sheets. So these are the tertiary structure in which there is a folding and coiling of the protein and which lead to the more complicated structure. We will call it as a tertiary structure. And in quaternary structure, there are more than one polypeptide chains are present, and it will lead to the formation of quaternary structure. Basically, hemoglobin is the best example of the quaternary structure in which there are monomers are present. This is basically tetramer in which different polypeptide chains are present, and it will lead to the quaternary structure. So I told you enzymes are nothing but these are the biological catalysts which catalyze the biological reaction. So if substrate is present, enzyme catalyst, then it will catalyze and leads to the formation of product. And the extent to which an enzyme is limited. So in like if an enzyme is made to catalyze one reaction, so it will catalyze that reaction only. It is not like it will catalyze any of the other reaction. It will specific to a one single reaction, so that is called as the specificity of the enzyme. And in human body, superoxide dismutase and catalase, these are the two fastest enzyme which will protect the body as oxidative stress. So superoxide dismutase and catalase. Superoxide dismutase, this you know na, oxygen on oxygen, if one negative charge is present, we will call as superoxide. So superoxide is harmful for human body. If in mitochondria superoxide is forming, then body has to protect itself from superoxide. So in that case, superoxide dismutase is an enzyme which is already present in the human body. At that in that case, the superoxide dismutase will activate and scavenge this produced superoxide, and the superoxide dismutase will then convert into the hydrogen peroxide and oxygen. And if hydrogen peroxide is formed in the human body, if it is in balanced amount, then it is okay, but if it is in excess, then it is also harmful for human body. In that case, catalase will activate, and catalase will convert hydrogen peroxide to water and oxygen, and these are the neutral elements which is required for the human body. So in that case, to protect the human body, these two enzymes will activate, and these are the fastest enzyme which are present in the human body. So in these enzymes, many inorganic ions and mainly metal ions are present. So metal ions are, if metal ions are present in the active site of enzymes, we will call it as metalloenzymes. So enzymes are further classified into these categories: oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. So oxidoreductases are the enzymes which helps in the oxidation reduction reaction in the human body, and transferases are the like enzymes which transfer one amino group like from one substrate to the other substrate. Hydrolases. Huh. So hydrolysis are the enzyme which hydrolyze ester, amide, and phosphate groups, and lysis this loss of H2O. If two substrate will condense and it will um, like water and carbon monoxide will release. In that case, lysis will help in that reaction. And isomerization is isomerization of substrate, and ligases it will like help in the formation of a new bond between two substrate. So this is the enzyme specificity. I hope this you people will know. Enzyme specificity, as I told you earlier, this enzyme specificity, like how, like to which extent an enzyme is specific to a particular reaction, is known as enzyme specificity. So basically, in an enzyme, one active site is present, which is designed in such a way so that it will bind to that of the particular substrate. So so it will react with that substrate only. So we can see that this is the enzyme. The active site of enzyme is looking like the surface of an enzyme is looking like this. So if the substrate surface is matching with this site, only then it will settle at this enzyme's activate site, and enzyme substrate complex will form, and it will catalyze this reaction, and it leads to the formation of products. So these are the example of enzyme specificity. So there is no need to go in detail of this. So this is the uh, basic overview of this course, in which we are mainly dealing with we are like bioinorganic chemistry, in which we are dealing with some of the biological aspects and the use of ions in biology, human body, and these all things we will read in this course. And what is the role of proteins, enzymes, what are enzymes, and we will learn different enzymes, like the enzymes which are useful in the um, oxygen carrier, mainly hemoglobin, myoglobin. Hemoglobin is the enzyme which is used to like transport oxygen from one body part to the, like from it will carry oxygen to the different organs, whereas the myoglobin will store oxygen and will supply to the body whenever it is required. So other enzymes also we will read, like zinc based enzyme, copper based enzyme, iron based enzyme. So different biomolecules we will learn in this course. So these are the different books which we can refer to like which we can refer to learn this course. And there are some papers also. uh, so so there are some papers which also we can refer to read this course. So any doubt till this uh, any doubt?
Hello.
Yeah, yeah, Gajanan, tell.
Ma'am, I have one query. I
Hmm.
think it's off the topic,
No,
but
no issue.
ma'am, what is the commercial application of all this? Like
What is the
commercial application, ma'am. Like,
commercial, in which sense you are asking?
like it will be in supplement industry, like future scope. How do I phrase it?
This is a requirement of our body, right? So if it is not present in our body, like what is commercial? I don't think like there is commercial requirement, means I can't understand in which aspect you are asking. I am telling about biology aspect, right? I am talking about enzymes and all. I am not talking about drugs here.
Like it's like in vitamin industry, they mainly focus on supplements, na?
Yeah, okay, okay. In that aspect,
That aspect.
so right. In that case, what can do is, like if there is some deficiency of, what we will tell, if there is some deficiency of any enzyme in our body.
Like patient is anemic or something.
Huh. Okay, okay,
If patient is
so
anemic,
in that case, I means exactly maybe I won't be able to answer for this question, but in, like, according to my aspect, I will, like, I am also basically from that field only. So in our labs also, and in like a commercial aspect also, if there is some deficiency in human body, like if some enzyme will be deficient in our human body, like I am telling, I was talking about superoxide dismutase. So if superoxide dismutase, like it will be deficient in our human body, then like some drugs we will synthesized which will mimic superoxide dismutase, like some small molecules or some metal complexes which we will synthesize, that will mimic superoxide dismutase. So it will act as a supplement of superoxide dismutase. So in this way we can think about this course, that small molecular metal complex has to mimic of that enzyme. In that case we can like use it as a supplement. Am I clear?
Yes, yes, yes.
Yeah. So basically, like, to overcome the deficiency of a particular biological enzyme, we have to synthesize a small molecule metal complex, like some natural product or something which can mimic the active site of a deficient enzyme, so that it will act as, like, it will act like same as that of enzyme. Basically we have to focus on the active site of enzyme, like how we can design the active site of that enzyme, like, outside the human, like biological environment, like in industry or something, as you are asking commercially. So we have to design some small molecule which can mimic of that particular enzyme and helpful in uh, overcoming the deficiency of that enzyme. Okay. Any other question? Yeah, Kalpana, you can tell.
Audible, ma'am?
Yeah, you tell.
Ma'am, will you explain classification of metal ions?
Can you tell again, please?
Metal classification of metal ions, ma'am.
Classification of metal ions. Kalpana, can you please write your question text in textbook? Like, classification of metal ions, like what you are telling, like the different metal ions present in different enzymes, like that you are asking. Can you repeat, Kalpana, please? Elaboration of metal ions, like what you are telling. I did not understand your question.
Hello, ma'am.
हाँ.
Metal ion, will you explain metal ion transportation, transport and storage process?
Metal ion storage and transport you are telling.
Yes, yes, ma'am. Yes, ma'am.
Okay, understood. This story, like this you are telling.
Yes, ma'am.
Okay. So in this case, what happen is like, basically you people know that cell membrane. Cell membrane is basically made up of phospholipids. So these are the this is the Bilayer cell membrane is the bilayer membrane of phospholipids. So these are the phospholipids which are connected to one another in head to head and tail to tail fashion. These are the polar groups and these are the non-polar groups. These are the hydrophobic groups. Huh. So these are the hydrophobic groups. So hydrophobic groups are connected in this way. So this is the bilayer membrane of phospholipids. So in this case, if the metal ions are present, so there are two ways which that metal ion can enter into the human body. So first they have to enter through the cell membrane, right? So cell membrane. So there are different type of transporters present on the cell membrane. Like this, you can see different channels, or we can say the different chaperone proteins, like different transporters. They are present on the cell membrane. But basically we all know that there is always a diffusion. What is diffusion is like when there is a concentration gradient across the cell membrane. You see, if there is a high concentration on one side and low concentration on the other side, then diffusion will happen. Basically, in diffusion, what happen is the metal ion will cross from high concentration to the low concentration. So this concentration gradient is helpful in transporting the metal ion from outside of the cell membrane to the inside of cell membrane. Basically, it is happening in the case of sodium potassium pump. In sodium potassium pump, what happen is there is the generation of concentration gradient which will helpful in the transportation of metal ion. But in some cases passive diffusion will help. In passive diffusion, what happen is different transporters and different proteins are present on the cell membrane which will act as a carrier. Basically it will act as a carrier to transport the metal ion from outside to the inside, or from inside to the outside. So these type of diffusion will help in the human body. It depends upon the protein to protein, like how it will like it will act in the biological system. Kalpana, am I clear?
Okay, ma'am. Thank you. Ma'am, will you explain trace elements and its roles in the bioinorganic chemistry?
Trace.
Trace elements. Trace elements.
Trace elements you're asking, right? Hmm. So you can see these are the bulk elements which are present in the human body. So there is no need to tell the role of this, because sodium, potassium, calcium, all we know, all these are very important. So these are the trace elements which are present in the human body. Out of this, you can see, if I will tell magnesium, silicon, fluoride, iron, zinc. These are the like trace elements. I won't be able to tell the role of all, but according to my knowledge, if you will see, iron is one of the trace element. But iron is very necessary for the human body as it is present in the active site of hemoglobin and myoglobin. If iron is not present in the hemoglobin, then there is no RBCs and there is no human life. Along with like myoglobin also, which is required to store the oxygen whenever it is required so that it can give it to the human body. So iron is basically one of the most important trace element which is present in the hemoglobin, myoglobin, and along with hemoglobin and myoglobin many other biomolecules are there in which iron is present. So zinc, uh, if we will see zinc, so carboxypeptidase and carbonic anhydrase. So the zinc is present in the carbonic anhydrase and carboxypeptidase. So uh, carbonic anhydrase is one of the enzyme which will convert bicarbonate into the water and carbon CO2. So this is also one of the important metal which is present in the which is present in the uh, biomolecules. And then copper is also which is necessary for human body, like it is involved in the various electron transfer reactions, um, and other you can see from here. I don't think here they are all. So basically these three are most important elements, and these are the most important elements which are present in the biomolecules, like iron, zinc, copper. And along with that, I here now I won't be able to tell the role of all these elements, but iron, zinc, copper, these are the trace elements, and these are the elements which is present in the most important biomolecules or enzymes, which is required for our body. Yeah, Kalpana, am I clear?
Thank you, ma'am. Thank you, ma'am. Thank you. Clear,
Yeah,
ma'am.
yeah, yeah. Okay, okay. Any other?
Ma'am, can you explain
Can you?
ammonia?
1 minute, you are not audible, Lakshya.
Ma'am, can you explain the hydrolysis of urea to ammonia by urease?
Hydrolysis of urea to ammonia
By
by
urease
by
enzyme.
urease. Okay, I can explain, but for that I also have to see. I will see if I will be able to answer this. Urease. Okay, so this is the urea. Okay, I have to check, Akshay. For means I can tell means not properly, but urease is a nickel-based enzyme, right? Actually, I have to check, Akshay. Can I tell this in the next class? First, I have
Okay,
to
okay.
check the like. I don't remember the structure or some. I don't know about this urease. I have to check what is in its catalytic site and how it is catalyzing urea to carbonic acid. From here, I can just overview that, like how it is acting. Like it is catalyzing with the help of water. Water is like attacking here, and it will leave. But as it is like bioinorganic class, so I have to first check the active site of urease. So can I tell in the next class?
Okay, ma'am. And one more question. Can you
Yeah.
explain the mechanism of sodium potassium pump?
Okay, okay. Sodium potassium pump. Okay, okay. Yeah, that I can
It's
explain then.
working.
Haha.
It's working. How it works like that?
Okay, sodium potassium pump. Actually, I have. Okay, I will see. I was having one slide for that. I have to check where it is. 1 minute. 1 minute. Okay, I'm telling. Or Akshay, my screen visible?
Yes, it's visible.
Yeah, yeah, yeah. Laptop is hanging. Hmm. So this is sodium potassium pump. Huh. So sodium potassium pump is one like membrane protein. It is present. You can see this is the phospholipid bilayer membrane. So sodium potassium pump is present. It is embedded in the membrane. So it is helpful in the transportation of sodium and potassium across the cell membrane. As we know that which is bigger inside, sodium or potassium? So sodium is smaller in size, potassium is bigger in size. So it exchanges the metal. So it exchanges these ions, sodium and potassium, in the ratio of 3:2. So what is happening is sodium will come across the membrane in the ratio of 3, whereas potassium will go as 2. So this is the protein, this is the uh, active site where 3 sodium will bind. So when these 3 sodium will bind, in that case this membrane will phosphorylate. So when this will phosphorylate, ATP will convert it into ADP. So when ATP will convert into ATP, energy will release, which will help in the transportation of sodium. Um, so sodium will help, it will help in the transportation of sodium. So when sodium will transport it, it will go here, and then expelling sodium, okay, and then potassium ejected. So pump will phosphorylate and it will change the conformation, which will result in the expel of sodium ions. So when sodium ion will go there, now there is a concentration in this pump. Concentration gradient is important. So when it will go, so then there is a concentration gradient between sodium and potassium. So when sodium will go there, the potassium will bind to the protein, and it will when potassium will bind, it will restore into the original conformation. Then that extracellular potassium which is present outside this cell, it will come inside the cell with the help of this conformational change. Basically in this case there is a conformation change of this transmembrane protein, and this is basically happening due to the concentration gradient. Akshay, am I clear? I have to explain again. I think I have to explain again. Hmm. So, so this is the sodium potassium pump. Okay, so in this what happen is this is sodium potassium. Oh my god. Huh. So this is the sodium potassium pump. So this is the pump which is embedded in the cellular membrane. So the cellular membrane, as I told you, this is bilayer membrane of phospholipids. So this sodium potassium pump is present um, inside this like cellular membrane. What happens in this case is then ATP will bind to the pump. When ATP will bind to the pump, ATP will phosphorylate this protein. When this protein will phosphorylate, um, this protein will phosphorylate, then ATP will convert into the ADP, and this phosphorylation of protein will facilitate the binding of sodium to this channel. So when these three sodium will bind to this channel, what happen is there is a change in the conformation of this protein. Now this the mouth of this protein, you can see it is in this side. When there is a change in this conformation, the mouth will open this side, and there is a transportation of sodium ions. So this sodium ion will go this side. Now there is this conformation change will help in the binding of potassium ions here. So when the potassium ions will bind, so these potassium ion will trigger as you know, there was phosphorylation. So when this potassium ion will bind, it will release the phosphorylation, means now there is no phosphorus atom. So this change in conformation happened because of the phosphorylation. So when there is no phosphorylation, means when there is a release of phosphorus atom due to the attachment of potassium, the protein, what protein will do, protein will again regain its original conformation. So when this will regain its original conformation, um, you see this mouth will open inside the cell, and this will trigger the potassium ejection. So the potassium like ion will eject inside the cell membrane. So like this sodium potassium exchange will happen, but this will happen in the ratio of 3 is to 2. So the enzyme, we can see that enzyme active site is designed is designed in this way, so that there is a space of 3 sodium and 2 potassium. And also on the basis of size also we can think that sodium are smaller in size than that of the potassium. So potassium will ejected out inside the cell in 2 number, whereas sodium will be in 3 number. So it will be the transportation in the ratio of 3 is to 2.
Yes, ma'am, understood.
I think now it is understandable. Okay.
Hello.
Haha, yes.
Sorry.
Haha, yes, Gajanan, today.
When will be the next class, ma'am?
Next class will be on Monday.
Okay.
Monday 6 to 8. I think your electrochemistry class has started, right?
Yes, ma'am.
But why there is a clash of classes then? Like you can attend one class only, na, at a time. I don't know. Means you people have, I think, lot of classes to attend.
Yes, ma'am.
Okay. So now, any other questions?
No, ma'am.
But I have to take class for 2 hours only. But I will do. If you have no question, then also, then I will ask some of questions.
Ma'am, the message has been sent that from 7 to 9 also there is class today.
Oh, means
Yes, ma'am. 6 to
me
2 slots at 6 to 8, 7 to 9.
6 to 8 and 7 to 9. So 7 to 8 will be bioelectrochemistry.
Bioelectrochem. No, ma'am. They posted in the mail now. That is only for bioinorganic chemistry. Second session is 7 to 9 and 6 to 8 is third session, ma'am.
No, no. Again, you tell again. There is a class of bioinorganic chemistry.
Yes,
Okay,
yes, ma'am.
okay.
So no, no, no. I am asking. I don't know. So again, there is a class of bioinorganic chemistry.
Bio.
Uh huh.
I think bioinorganic chemistry only, ma'am.
So who will take that class?
We don't know, ma'am. Session 2 is 7 to 9 for the bioinorganic chemistry.
Oh
But we
my
Today we
attended
got
god.
the mail.
Oh
session.
my
Yes.
god. So no, they will not inform like who will take the class.
We don't know, ma'am.
Oh my god. This is like I don't know how these classes. If these classes will clash, then I won't be able to take for 2 hours.
Ma'am, will you explain the important questions, how to cover the questions in the NPTEL?
How to cover the questions in NPTEL?
Yes, 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 ma'am.
Means you are asking for exam purpose.
Exam purpose only, ma'am. Yes, and major
From
questions
first
like,
class, you are you are worried about exams.
ma'am, important topics from the
Actually, what I felt is I
yeah.
also opted this course, but not from NPTEL. I opted this course from my institute, like from ISC only. So, from my perspective, if I will say, like the oxygen carrier proteins like hemoglobin and myoglobin, these are the most important topics to cover actually. So we have to means we should know like how these proteins are carrying oxygen from one to another. So hemoglobin, myoglobin.
Okay.
Then there will be if you I so people are joining now also. I don't know what they're doing. Okay.
Okay, okay, ma'am. Okay,
Ha,
ma'am.
so oxygen based iron containing, like if you will see there is a oxygen carrier. So in oxygen carrier also there are two categories. The first is iron containing proteins and the other is non iron containing proteins. So in that iron
Hmm.
containing proteins, hemoglobin and myoglobin are the most important topic, and the
Okay.
non iron containing proteins like hemerythrin and hemocyanin, these two are important enzymes. So these
Okay.
are the iron-based enzyme, and if we will go to the zinc-based enzyme, then carbonic anhydrase and carboxypeptidase. These two enzymes are very important. We should know each and every perspective of carbonic anhydrase and carboxypeptidase. And after that, zinc, the other which we discussed, trace element. What was the other? Iron, zinc, and iron sulfur clusters, redox proteins, electron transfer reagents, ferridoxins and rubridoxins, like iron sulfur clusters. That should be covered. That should be covered
Okay.
properly. These are the important iron sulfur clusters. And other zinc based enzyme, copper based enzyme. In copper based it will be ceruloplasmin and all these, and then vitamin B12 deficiency and excess of these metals. So these are the important topics for this course.
Okay. Okay,
Hmm.
ma'am. Thank you.
Mm-hmm. Okay, so you have any other doubts? You people have any other doubts? Now, what shall I do? I don't know. What shall I do? Actually, my class is from 6 to 8, and I prepared some slide for you. We discussed the doubt, and now I was thinking we will discuss some questions. So, I don't know. I have to continue or what I have to do. So now I can see there are 6 people, right? Kalpana, you also have class.
Yes, ma'am. I mean, yes, only.
Oh, you are in other
Ah,
class also.
yes, ma'am. Yes, ma'am.
Two classes simultaneously you are attending.
Bioelectrochemistry, they are allotted 9, ma'am.
So the other one is bioelectrochemistry or bioinorganic chemistry.
So tomorrow also so one session is there, no problem, ma'am. We continue, ma'am, no problem.
Okay. So, okay. What we will do, some 15 minutes we will read, then I will wind up. Okay.
Okay, ma'am, no problem, ma'am, no problem. Carry on.
Uh, so, okay. If you have some doubt, then you can ask. I will try to convey, I will try to clarify your doubt. Okay, then we will do some questions. Okay. I made some slides for question answer, then we will do that questions only.
Sorry for the
Huh?
disturbance, ma'am.
What? You were,
Hello.
yeah.
Ma'am, they given one question in the assignment in NPTEL, ma'am.
Uh-huh.
Which enzyme is, turns to apple and potato turns to brown color? Is it tyrosinase enzyme or polyphenol oxidase enzyme?
Again, you ask question or you can write. Can you please write?
It's
Ma'am, is
tyrosin.
it tyrosinase?
Can you ask the
Hello.
question? Uh-huh.
Which enzyme turns to which enzyme turns apple and potato to brown color, ma'am?
I think it is tyrosinase.
Tyrosinase.
Hmm.
Okay, okay, ma'am.
Hmm.
I confused just so that's why I'm asking, ma'am. Okay.
Hmm. I was having some slide also for this actually. Apple also will turn that white to yellow, right? Okay, then we will discuss some questions. I have made some slide for you people. So, deficiency of which metal causes pernicious anemia?
Cobalt, I guess.
Do you know?
Iron,
Uh-huh.
iron,
Iron.
iron.
Iron is not in the option.
Cobalt.
Yeah, I think cobalt is the right answer. So pernicious anemia is pernicious anemia is vitamin B12 deficiency. It is different from anemia. If it is like causes anemia, then it will be iron, but it is pernicious anemia which is due to the vitamin B12 deficiency. So if vitamin B12 contains cobalt in its active site, so when there is a deficiency of cobalt, there is no biosynthesis of vitamin B12, which leads to the pernicious anemia in human body. So the next question
Man,
is,
are you sharing your screen or
oh, I am
simply?
sorry. No, I was sharing. I'm sorry. Sorry. Okay, that's why I think you did not you didn't see the
Yeah, now it's
options.
visible.
Ha. Right. Now you can see, right?
Yes.
Yeah. So this is pernicious anemia. So pernicious anemia is vitamin B12 deficiency anemia. So the next question is, which of the following are the examples of iron sulfur proteins? I think this has
Rubidoxine. Rubidoxine.
rubridoxine. Yeah, rubridoxine is one of the iron sulfur clusters. And the other, methane monoxygenase, risque protein, aconitase. So rubridoxine, risque protein, aconitase. These all three are the iron sulfur clusters. You see, this is the rubridoxine in which iron is present, and sulfur is bounded to the iron. This this is aconitase and this is the risque protein. So in all these three are the iron sulfur proteins. So the rubridoxine, risque protein, and aconitase, all three are iron sulfur protein. Actually, the question should be which of the following is not iron sulfur protein. So okay, fine. So the hemoglobin is a monomer, dimer, trimer, or tetramer.
Tetramer.
Yeah, tetramer. So hemoglobin, this is the hemoglobin. So we can see this is the tetramer of this. This is the monomer of hemoglobin. So if this monomer will like in hemoglobin, so these 4 units are present. So this is the tetramer. So which of the following is a copper transport protein? So you can see na hemoglobin is what? Hemoglobin will not transfer protein na. Hemoglobin is iron containing protein. So myoglobin is also. So these two options got cancelled. So now out of ceruloplasmin and plastocyanin you have to see. So plastocyanin is an copper containing protein, but it will like what I will tell. It is involved in the electron transport reaction. So for now, ceruloplasmin is one of the enzyme which will transport copper in the human body. So the next question is most abundant element by atomic percent in human bodies. This you should answer actually.
Hydrogen.
Oh my god. Now only I thought. What?
Oxygen, ma'am.
Yeah.
Oxygen, ma'am.
Yeah, oxygen is the most abundant element which is present in the human body. You see everywhere it is oxygen. Water, oxygen. In mitochondria also oxygen, superoxide, hydrogen peroxide, oxygen is releasing. So everywhere the most abundant in human body also it is oxygen which is the most abundant. So the metal ion used in the MRI contrast agent. So MRI, what is MRI? MRI, you know now we are going to the doctors to do MRIs.
Yeah, they do scanning with that.
Yeah, they do scanning. So what is the full form of MRI?
Magnetic resonance imaging.
Imaging, right. So this is the magnetic resonance imaging. So one of the rarest element is used in MRI, which is F block element. So which is gadolinium. Gadolinium is used in the MRI. So this is the metal ion which is used in the MRI. So, in the active site of many enzymes, metals are coordinated by the amino acid histidine. Which element in histidine donates the electron that form the coordinate bond? So, I am expecting you people know the structure of
Nitrogen.
histidine. Yeah,
Nitrogen, ma'am.
yeah, yeah. So, this is the structure of histidine. So, nitrogen is present with lone pairs. So, the nitrogen is donating the electron to form the coordinate bond. So, what is the geometry of cisplatin? Cisplatin, you people know, right?
Square planar.
Haha.
Square planar.
Yeah, cisplatin,
Scorpion.
you know what is cisplatin, where it is used?
For cancer
PTCl4.
treatment.
Yeah,
For cancer treatment,
yeah,
personally.
yeah, yeah. PTCl4 2 negative and it is used in the cancer treatment. So cisplatin it is having square planar geometry, right? So which enzyme is responsible for spontaneous degradation of red blood cells? What is red blood cell? Basically, it is RBCs, right? It is hemoglobin. Hemoglobin means in hemoglobin I taught you today hemoglobin for that heme is required. Okay, so if heme is required in the body, but if any enzyme is biosynthesizing in body and if its work is done it also has to degrade, right? So what is the enzyme which is responsible for the degradation of red blood cell? Name itself is indicating which enzyme. You read all the 4 names of enzyme. I think name itself is
Ma'am,
indicating.
hemioxygenase.
Yeah, heme oxygenase. If heme oxygenase, heme oxygenase will convert heme to biliverdin, ferrocyan, and carbon. Basically, what heme oxygenase is will do, it will degrade the hemoglobin and convert into the different like things like carbon monoxide, ferrocyan will release, biliverdin will form. So the name itself is indicating that heme oxygenase will degrade the RBCs in human body. So the ground state spin multiplicity. This is very simple question. We saw in human body the most abundant element is oxygen. So they are asking for ground state spin multiplicity of dioxygen. It is very simple, right?
Triplet, ma'am.
Triplet, right? Triplet it is. How it is triplet? You can explain, right?
Ma'am, there is two unpaired electron
Yeah.
in
Hmm.
pi antibonding orbit.
Yeah, yeah, yeah. So there are two. electrons present in the, so it will be 1s2 2s2. What is the atomic number of oxygen? 8, right? So it will be 1s2 2s2 2p4. So if 2p4 is there, it will, I have no pencil for writing in laptop actually, so my handwriting here is very poor. Okay, so it will be 4, 1 2 3 and 4. So here, two unpaired electrons are present. So for calculating spin multiplicity, the formula is 2s plus 1. So what is the spin here? Half plus half, 1. So 2, 1 plus 1. It will come out as 3. So triplet is the ground state spin multiplicity of dioxygen. I think I am done with these questions. I don't think I have. So I'm done with this question. If you people have any doubt, then you can ask. These questions are very easy. Simply I made these slides to. I thought we people will interact, then it will be good for you, and you will ask some doubts. And if you have some other doubts, then you can ask. I will try to clear. Still, half an hour is there. If you want to discuss something, then we can discuss.
Ma'am, that hemoglobin is tetrameric unit, right? But tetrameric unit of exactly what? Is it myoglobin itself or something else?
No, no, myoglobin itself. What is that? Like what you see in myoglobin, first we will not consider hemoglobin. First we will go to the simple one. So what is myoglobin? Myoglobin is the monomer. And what is myoglobin? Myoglobin is the monomer in which heme unit. Okay, I will show you. This is basically myoglobin. But here, so this is basically, you don't see this one. You see this one. You focus on this one. You focus on this one. So this is the heme unit. Heme unit in which this is the pyrrole units. But I will tell this is I pyrrole. No, no, no. Some ring we will tell now. I for
Porphyrin ring.
porphyrin ring. So this is a porphyrin ring in which iron is bounded, right? So one oxygen will come which will uh, which is binding to that iron, and the polypeptide chain will be at the sixth coordination site, right? So it will be like some all the six coordinating site will be fulfilled. So that is the myoglobin, in which four sites will be occupied by the porphyrin ring, fifth site will be occupied by oxygen, and sixth site will be occupied by the polypeptide chain which is present in the myoglobin. So this will be the myoglobin. This is the monomer. This is the like exact structure of the myoglobin. And if this unit will repeat itself in four manner, means four, what I will tell, four means, you understand na? Four things, then it will leads to the formation of hemoglobin. So it is basically the tetramer of the myoglobin. Understood?
Yes, yes, ma'am. Like
So
I had doubt whether it was exactly myoglobin or was
no,
there
no,
any structural
no,
change?
nothing, nothing. In myoglobin, if we will see, one iron atom is present, one porphyrin ring is present, and one oxygen if it is binding, and like if it is inactive myoglobin, then weak ligand like water will be bounded, and if oxygen has to bind, like oxymyoglobin will form, then oxymyoglobin, and the sixth side will be occupied by the polypeptide chain. Now this polypeptide chain, I told you, like it is very complicated. This polypeptide chain itself will contain 2 alpha and 2 beta units. So this full will form myoglobin, and if this will repeat in 4, like, then it will form
Yes, yes,
hemoglobin.
yes.
Okay.
Yes, ma'am.
So like this, it will be the tetramer of the myoglobin. Any other thing? Do you people have doubt? Where you are? Uh, how many? Akshay, Kalpana, Nayan, you people have some doubt, then you can ask. So, what you people want to read in next class? It will be on Monday, 6 to 8. So, if you people will tell like this topic we want to read, we have some doubt, we need more clarification regarding this topic, then you people can tell. I will like prepare accordingly. Means I can discuss assignments also. I can discuss different question. I can discuss your doubts. Anything is fine for me. But if you are having some specific doubt, like this topic we want to clear more, then you people can tell. Like if you have
Ma'am, that cobalt mechanism, that urease
urease,
mechanism,
urease. Huh.
that one, and that redox stability of water, like under this pH, that it was kind of electrochemistry thing. I was not able to get that.
Okay, I will write here what you are telling. Urease catalytic cycle. And what other thing you told?
Redox stability of water, like,
Redox stability of water.
yeah, like during that electron redox active metal ions in enzymes, they were speaking about specific volts between them. They are
Okay, I
stable
think
pH.
I will show you one slide. You're talking about this. 1 minute. I don't know where it is now. You are telling about oxygen plus one electron will give superoxide, then hydrogen peroxide, right? Like this you are telling, right?
Uh, no, not that. Like, uh, why metal ions, certain metal ions, has been chosen as redox center. Like, they spoke about the pH range in which water is stable,
Which
where they
water?
use Nernst equation and different pHs. Then overpotential concept also comes there.
1 minute. I don't know what you're talking. I think maybe in that third lecture it will be there.
Yeah, yeah.
You are telling about this, Akshay.
What no?
And my slide is visible to you.
Yeah, yeah, yeah. It's visible.
You are talking about this. Like
No, not this.
how it is forming water.
This one only. Yes, oxidation of water.
This is reduction of oxygen, right?
Yeah, yeah. Oxidation of water.
Hmm. So you are
Like
talking.
they had used that graph, no, ma'am. Like between 4 to 9 pH, uh, like there is over voltage, and above this, water will be oxidized to water. Below this, it will be reduced to
Okay,
hydrogen.
he has taught you this.
Yeah, he told it should be not. It should not be below this, so that it won't get reduced to hydrogen in our body.
Oh, I don't know. I have to see the lecture. In which lecture it is? Can you tell me? We will just see, right?
It's in the first week lecture, I guess.
This.
I don't exactly know the lecture
No, that
number.
is fine. We can just check which you are talking about. Then only I will just see and explain.
It's the fourth lecture, first week.
It is in lecture two.
First week, fourth lecture.
Okay, once I will check. Okay, Akshay. Once I will check.
Okay, ma'am.
Okay, some graph is there. Is it?
Yeah, it is all about EMF and
No
EMF.
audible.
It's about electrochemistry kind of.
You are not audible, Akshay.
Now am I audible?
You are not audible. I think you have to write. EMF things come. EMF. Okay. Okay. Akshay, I will see. I will just see. Then I will let you know. In next class, I will take care of these two things. And yeah, okay, okay, nothing. So, okay. So, okay. So now there is nothing to, I think, discuss. So, can we wind up the class? Is there any
Yes,
doubt
ma'am.
or something?
Yes, ma'am.
Any other topic you want me to clear for next session? Then also you can tell. Okay, okay. So, okay, we will discuss in the next class. Hmm? Now you people can continue with your other classes or whatever you want to do. Okay, bye bye. Bye. Oh, Kalpana, you told you want to tell something? Kalpana has
No,
raised a
no,
hand, but
no,
Kalpana, you are
ma'am.
not audible to me.
By mistake, it's came, ma'am. Sorry. No, ma'am.
Kalpana, you are not audible. You want to tell something?
Nothing, ma'am. Nothing.
You can write actually, you are not audible. Nothing is, means nothing I can hear. Kalpana, I am not able to hear you. If you have something, then you can write. I will read. Okay, okay, okay. Actually, it is showing that Kalpana has raised hand, so I thought you have some query. Okay, then Kalpana, bye bye. I'm leaving the class, so we will meet next Monday. Okay, okay. Welcome, welcome.